Good morning. Microbiology. What does microbiology study and why is it important? Microbiology is the science responsible for the study and analysis of microorganisms, tiny, living things not visible to the human eye. And what is the importance of microbiology? In the field of health and medicine, microbiology is of the great importance since it is in charge of studying pathogenic microorganisms such as fungi, viruses, parasites, and bacteria that can cause diseases in humans. Okay, there are two types of microbiology, and they are basic microbiology and clinical microbiology. Okay, basic microbiology studies the biological, ultrastructural, biochemical, and genetic aspects of microbes. Okay, the clinical microbiology diagnoses and characterizes pathogenic microorganisms isolate from patients for diagnostic and or epidemiological purpose. And in this case, you will see basic microbiology. Okay, and the topics are the microscope, cells, difference between cells, growth and metabolisms, along with topics on control of microorganisms. Microscope. Okay, where is the microscope? Microscope allow us to obtain magnificent images of objects in order to detail, study and analyze them. In this sense, they are used in various areas of knowledge, such as biology, medicines, archaeology, etc. The microscope was invented in 1590 by a lensmark named Zacharias Jensen. Microscopes in general are composed of the following parts: the eyepiece, through which we can see, the lenses, which are above the objective, the stage, which is a small platform where the object holder contains what to observe is placed, the focus that serves to focus object, and the condenser, which serves to concentrate the light on our object. Since the invention of microscope, there have been important advances in science and medicine. It was instrumental, for example, in the discovery of cells, protozoa, bacteria, spermatozoa, red blood cells, etc. Now, types of microscope are electron microscope. An electron microscope is that uses electron radiation instance of light to form images of objects that are very small to the naked eye. Opticals. And the other is the optical microscope. And the optical microscope, also called light microscope, consists of a system of optical lenses that uses visible light or photons to magnify the images of the objects observed. Stereo microscope. The stereoscopic microscope is characterized by displaying an image in 3 dimensions. Okay, the simple microscope. The simple microscope is one that uses a convex lens to magnify the images of the objects observed. Compound microscope. A compound microscope is a microscope with more than one objective lens. Okay, and that is about a lot of things about microscope. So we'll continue with cells. Okay, cells. In biology, it is said that the cells is the functional and structural unit of all living beings because all cells are capable of performing the functions of nutrition, relationships, and reproduction. And it is important to say that all organisms are formed by cells. Examples of the difference of two major cell types are the prokaryotes and eukaryotes. Examples of prokaryotes. Bacteria. Bacteria are unicellular organisms that belong to the group of prokaryotes. This means that they lack cell nucleus and organelles. Organelles such as mitochondria, chloroplast, or the Golgi apparatus. Golgi apparatus, so their genetic material, DNA, is found free in the cytoplasm. Example of eukaryotes, fungi and yeast. Eukaryotic microorganisms are not only larger and structurally more complex than prokaryotes, but also prokaryotes have a different evolutionary origin. The eukaryotic domain is composed of algae, filamentous fungi, and yeast, mucus fungi, and protozoa. Algae are distinguished because they contain chlorophyll and can perform oxygenic photosynthesis in their chloroplast. They can be unicellular or form aggregates. Also, most are microscopic. Some can grow several meters along. Other types of microorganisms are parasites. Parasites. In biology, the term parasite refers to an organism that grows and feeds at the expense of a different organism called a host. A different organisms, the parasite obtains nutrition and physical protection without making any contribution to the survival of its host. And viruses, even smaller than bacteria or yeast, there are biological entities. Now, as viruses are not considered cells because they lacked many of the attributes of cells. Okay, difference between cells. Bacteria are microorganisms that can have different shapes. They can be spherical, elongated, or spiral. There are harmful bacteria called pathogenic bacteria, which cause diseases. But there are also good bacteria. Microorganisms are classified into four groups: bacteria, viruses, fungi, and parasites. Each of these groups has different aspects regarding their relationship, structure, morphology, nutrition, and reproduction. Their difference between the groups is due to a greater thickness of the cell's wall, especially in a layer of gram-positive bacteria, where Eden and I precipitate and stain the wall violet. In contrast, in the group of gram-negative bacteria, the crystal violet dye is usually lost. However, there are some bacteria in which the method of identification cannot be applied due to the high lipid content of the cell wall layer. We will continue with growth and metabolisms. Uh, Metabolism or catabolism or destructive is the process that produces the energy necessary for all the activity that takes place in cells. Metabolism is the set of chemical reactions that take place in the body cells to convert food into energy. Our body needs this energy for everything we do, from moving to thinking to growing. Okay, what are the cellular metabolisms and what are its phases? Biological metabolism is composed of two conjugate phases or stages. And now, as catabolism and metabolism. The first is concerned with releasing energy by breaking given chemical bonds. The second with using that energy to form new chemical bonds and compose new organic compounds. Okay, continue with control of microorganisms. What is bacterial control? Control of microorganisms in vivo means the administration of bactericidal and bacteriostatic agents to a living being. Uh, Antimicrobial agents are substances administered to a patient to reduce the microbial load or prevent the growth of microorganisms without causing harm. How can bacteria be controlled? Antibiotics are drugs used to treat infection caused by bacteria. They work against infection by killing bacteria or hindering their development and reproduction. But it should be remembered that they are not effective against viruses, fungi, or parasites. How can bacteria be killed? Antibiotics kill bacteria. When antibiotics are not used properly, they may not kill all bacteria, and bacteria that the antibiotic does not kill become stronger. Okay, they are said to be everywhere, but we cannot observe them with the naked eye. Um, they exist even before the formation of the Earth's atmosphere, and today we depend on microbes for uh, to obtain various useful products for food, pharmaceuticals, and other areas. The microorganisms are classified according their shapes, size, or properties, and can be both beneficial and can be detrimental to other living things. Okay, thank you.